So it is a really great pleasure to introduce Victoria Ryg-Cornejo today from the Division of Infection and Immunity. Victoria joined my lab in 2010 when she was actually coming back home from finishing her Bachelor in Biology degree at the University of Montpellier in France. In 2010, Victoria started honors, and she had a very successful honors year. Her work on crosstalk between natural killer cells and dendritic cells resulted in a publication, which was then followed by a second publication that was a methods paper. In 2011, Victoria decided to start her PhD. And that was interesting timing because that coincided with a big shift of direction in the research areas in my lab. So after 10 years of working in cerebral malaria, my lab started to focus a lot more on mechanisms of immunity to malaria. So before starting her PhD, Victoria had to set up new infection models. She had to set up a whole bunch of new different techniques and generate new tools before she could even start addressing the questions of her PhD work. So all that hard work was actually instrumental to establish the foundations of a new research area in the Hansen lab. And today, Victoria is going to tell us about some of the findings emerging from that research on germinal central responses during severe malarial episodes. Victoria.
Alrighty, thank you for that lovely introduction, Diana. Hopefully I can do that all justice. So I'm going to dive straight in and just show you this map, which comes from the Second World War, and it's called a news map, and it was used by the Allied army to communicate to soldiers as they fought around the world of various dangers. And this particular map was illustrated by Dr. Seuss, as you can see by his characteristic drawing style up there. And it shows the spread of malaria at the time, and you can see that that was really quite significant, and it was found even in Northern Australia and Southern Europe. And fast forward 70-odd years, and you can see that really, despite really significant efforts of malaria control and eradication, it's still a really big worldwide problem, and it causes infection in up to 200 million individuals every year, with almost half the world's population considered to be at risk of infection. So malaria is caused by parasites of the genus Plasmodium, and there are 5 known species which infect humans, but Plasmodium falciparum is the one that causes the most severe forms of the disease. So the parasite has a fairly complex life cycle in the human host, which commences with the injection of sporozoites from an infected mosquito. And those sporozoites will go on to replicate within the liver, where they'll emerge as merozoites into the bloodstream. And those merozoites will set up a cyclic infection within the host erythrocytes for their asexual development. And a proportion of those can also go on to differentiate into gametocytes, which are the sexual form taken up by the next mosquito. And for the purposes of this talk, I'm going to be focusing exclusively on the blood stage of development. And in a minority of cases, that blood stage infection can lead to the development of really, really severe symptoms, and which can be grouped into different types of syndromes. And so here we can see a group that is related to the syndrome cerebral malaria. You can also develop severe anemia, which isn't listed here, and then more sort of general severe disease, such as organ failure or metabolic acidosis. So thankfully, the development of those severe syndromes can be prevented by the development of naturally acquired immunity in endemic areas. And one of the really significant questions that remains in the malaria field is how that immunity actually develops. And we know that it takes a long time to develop, and it requires repeated infections, which you can sort of see summarized in this graph, where you can see the development of different infection outcomes based on the age of the individual. And so because they lack substantial protective immunity, young children under the age of 5 will experience the highest incidence of the development of those severe syndromes I just mentioned. And the same effect is mirrored in unexposed adults and those World War II era soldiers. However, as they get older, those children will experience more infections, and they'll develop a level of immunity which leads them to only sort of tend to get febrile illness. And once they've reached adulthood, that level of immunity will be sufficient enough to allow them, to give them protection from symptomatic infection. But it's important to note that that immunity has never actually been found to be sterilizing. So although both cell-mediated and humoral responses have been shown to be important in protection against malaria infection, the highest protective effect has been attributed to the malaria-specific antibody response. And this was first addressed in this landmark study from the mid-1960s, in which hyperimmune serum from adults was passively transferred into children who were sick with severe disease, and it allowed those children to recover. Subsequently, those studies were repeated in both mouse models and primate models of the disease. And further studies showed that it was actually the associations between some specific malaria-specific antibodies with protection. So as Danica told us all last week, the development of immunity to malaria is a very complex process, and many different things can intervene to change that development of immunity. But in the development of malaria-specific antibody responses, it's the parasite itself that's been attributed to the slow acquisition of those responses. And that's because the parasite expresses a really large number of antigens across the different life cycle stages, but also those antigens are known to be highly polymorphic in certain cases, which means that it's sort of thought that it just takes the host a long amount of time before they develop a panel of antibodies sufficient enough to give them protection. However, more recently, attention has turned to the development of B-cell responses during malaria infection, whether those might be impaired in some way. And evidence has been found of direct immune modulation by the plasmodium parasites of B cells, so that, for instance, it's known to directly activate polyclonal B cell responses. In addition, when we look at the types of circulating memory B cells in people who've had malaria, you can find changes in those populations, including this induction of a so-called atypical subset of memory B cells, which are thought to have poor capacity to redifferentiate into antibody-secreting cells upon restimulation. And finally, the generation of B cell responses in and of themselves may be compromised, and that's what I'm going to focus on today during my talk. So there have been very few studies that have looked at the generation of malaria-specific B cell responses in human populations, and one which was undertaken in Mali in an area of high seasonal transmission where clinical episodes are quite frequent in young children, found that the memory B cell populations against two merozoite antigens, AMA1 and MSP1, which are very abundant proteins, found that those memory B cells were acquired at the time of the infection, but that at the end of the transmission season, those populations had contracted back down to just pre-infection infection levels. And as a consequence of this, the acquisition of memory B cells against those two antigens only occurred incrementally, and it only stabilized in adulthood. In contrast to this, when the same responses were measured in an area of low seasonal transmission in Thailand, where people experience only very infrequent clinical episodes, found that populations of memory B cells could be generated even in the absence of reinfection. And you can see that here, where the frequency of memory B cells against those same two merozoite antigens have remained stable, even if the last known infection was 50 months prior. And so taken together, this data has led us to hypothesize that severe clinical malaria episodes might be limiting the development of B cell responses. Now I'm just going to step away from malaria for just a moment and just to explain the complex processes that need to happen before a memory B cell response can actually occur. So because malaria is a blood-borne infection, the immune responses that occur against it are generated in the spleen, which can be broadly subdivided into two areas: the red pulp, which is rich in red blood cells, and the white pulp, which is enriched for leukocytes. And the white pulp can be further subdivided into segregated areas of B cells and T cells. So when an antigen is detected, those T and B cells will become activated, and they'll migrate to the border of the follicle where they can begin delivering signals to one another. And a portion of those B cells can go on to migrate out of the follicle and into the red pulp, where they'll form these foci of short-lived plasma cells. And those plasma cells can be really important for secreting large amounts of low-affinity antibody, which can be really important in initial pathogen clearance. The T and B cells can also go on to migrate into the follicle, where they'll set up these germinal center structures where clones of B cells will undergo rounds of proliferation and somatic hypermutation. And with the help of this specialized T helper cell subset called a T follicular helper cell, the highest affinity clones will be selected for entry into the memory B cell or the long-lived plasma cell compartment. And memory B cells are thought to re-enter the circulation where they act as a sentinel population so that they can really rapidly redifferentiate if the pathogen reappears again into high affinity effector cells. Long-lived plasma cells, on the other hand, take up residence in the bone marrow where they're thought to sort of constitutively secrete high affinity antibody, which can also be important if the pathogen re-emerges. So it's not clear in what way severe malaria infection might be impeding in those processes, but one hint has come from the post-mortem analysis of spleens obtained from people who died of severe malaria infections caused by Plasmodium falciparum. And so you can see here that that immunohistological analysis of those spleens show that there is a really significant loss of splenic architecture. And in particular, you see these B cell follicles that are present in the uninfected individuals, and those germinal centres are absent from those tissues, which might provide a hint as to how severe malaria infection is impeding the generation of B cell responses. Now, it's fairly common for malaria researchers to obtain peripheral blood samples to look at the circulating immune cell populations, but it's much more rare for us to be able to get samples like this to look at the processes underlying the immune responses. And for that reason, for the most part, characterization of those responses has occurred using mouse models of the disease. And historically, the most commonly used strain of parasite has been Plasmodium shibati. And infection with that parasite causes an infection which is resolved by the host within about 2 weeks. And because it's a non-lethal infection, it's been used as a model of human mild malaria. And so when mice were infected with Plasmodium shibati and their splenic architecture was analysed, disruption of splenic architecture was noted, but that was temporary, and germinal centres did eventually form, and B cell responses did occur. Which means that the phenomena that are seen here do not fully recapitulate what we saw in the human severe malaria infections, which makes it difficult for us to conclude, to draw any conclusions from this model about human severe malaria. And that brings me to the overarching aim of my PhD project, which was to determine whether or not severe malaria episodes were having a detrimental effect on the generation of B cell responses, and also look at the mechanisms by which that might be occurring. And to do that, I looked at a number of different B cell responses. So I looked at the plasma cell response during that severe malaria episode, as well as the germinal centre response, and I characterised some of the populations that were taking part in that. And finally, as I'll get to in just a moment, because inflammation is such an important part of severe malaria infection, I looked at whether that may have been impeding the germinal centre responses in some way. And because I wanted to specifically look at severe malaria, I used the mouse model of malaria, Plasmodium berghii. So infection with Plasmodium berghii causes the development of cerebral malaria within 6 to 10 days following infection, and that cerebral malaria is characterized by the development of neurological symptoms such as hemiplegia, ataxia, paralysis, coma, and finally death, which you can see in this survival curve here. And the mice die with relatively low parasitemias in that infection in a pathology that's characterized by dysregulated host immune responses, which I'll talk to you a little bit about now. So although the etiology of severe malaria isn't completely understood, we do know that during infection, parasitized red blood cells will sequester in the brain microvasculature of infected mice, triggering these localized inflammatory responses. And at the same time as that, populations of T cells and NK cells will be activated to start to produce a pro-inflammatory response in the spleens, and they'll start secreting cytokines such as interferon gamma. And those cytokines are thought to go on to activate populations of monocytes and macrophages, which will then begin secreting pro-inflammatory factors of their own. And although that inflammation can be really important to aid pathogen parasite clearance, in the case of Berghii infection, it sets up these positive feedback loops, which lead to really significant increases in the levels of pro-inflammatory cytokines. And one of the effects of those pro-inflammatory cytokines is to upregulate inflammatory trafficking pathways. So what we find is that CD8 T cells, CD4 T cells, and NK cells upregulate the CXCR3 chemokine receptor. And at the same time, in the brains, because of that localized inflammatory response, the ligands for CXCR3, CXCL10 will begin to be expressed. And as a consequence of that, those cells will infiltrate the brain, where along with the sequestered red blood cell, the parasitized red blood cells, will start causing problems like hypoxia, edema, and hemorrhages, which occur at the onset of severe disease. So because we wanted to study the effects of severe malaria infection and exposure to the parasite in the absence of all that host and parasite-mediated immunopathology, I compared infection with Plasmodium burgii to immunization with an irradiation-attenuated form of the same parasite. And for simplicity's sake, throughout the talk, I'm going to refer to the burgii infected mice as infected mice, and the mice that have been given irradiated parasites as immunized mice. So in order to ensure that both groups of mice were receiving the same antigenic load, the immunized mice would receive 3 doses of the attenuated parasites to mimic the increasing parasitemia that we see in infected animals. And because I wanted to look at the longer-term effects of that severe malaria infection, all the mice were given antimalarial treatment at the onset of early cerebral symptoms in the infected mice to prevent lethality. And in that way, we were able to establish a point of comparison between the development of immune responses in the absence of the clinical episode and also inflammatory responses. And to verify that there were indeed less inflammatory responses in our immunized group, I looked at the spleen homogenates of those mice early on in infection for the production of pro-inflammatory cytokines. As you can see by the bars in blue, which are the infected mice, compared to the bars in stripy grey, which are the immunized mice, I found that the production of both IL-12 and interferon gamma was significantly higher in the spleens of the infected mice at different time points following infection, showing that by exposing mice to attenuated parasites, we limited that inflammatory response. And in another set of experiments, I wanted to look at the role of plasma cell responses in these processes. And so when we want to look at whether or not plasma cells are important during an infection, we look for changes in the level of parasitemia and also survival of our different groups of mice. So this is just to sort of show you an example of how we do that. And this is an infection with Plasmodium schaubouti in wild-type C57 Black 6 and also BLIMP1 CD19 Cree mice, which specifically lack plasma cells. So when we infect those mice with Plasmodium schaubouti, which is that mild malaria version in which is non-lethal, we see this increase in parasitemia in the white wild-type animals, which leads to and resolution of that parasitemia, which leads to no problems with survival at all. On the other hand, when we look in our BLIMP1 CD19 Cree plasma cell deficient mice, we find that parasitemia goes uncontrolled, and as a result, those mice have significantly higher death rates. So now when we take our plasma cell deficient mice and we infect them with Plasmodium burgii, what we see is something quite different. We see that both wild-type and plasma cell deficient mice have similar parasitemias, but also that they have similar rates of death, which suggested to us that the plasma cell responses might be impaired during this model compared to this one, where they're both generated and participate in protection. And so I went on to look at the antibody responses in the infected and immunized mice at the time of the clinical episode, and I measured those antibody responses against that merozoite surface antigen, which was highly abundant. And I found that when you took away the clinical episode and the inflammation, you had improved unswitched and switched antibody responses, which further provided support for this model to be used to look at the effect of the severe malaria episode. So given that we saw all these sort of potential problems with the plasma cell response, we decided to take a closer look at the plasma cell populations that were generated during an immune response. The problem was that plasma cell populations are quite difficult to track in vivo. And that's due to the variability in the surface expression of a lot of the markers that are used to characterize them, which you can sort of see summarized in this table. However, the plasma cell lineage, as I sort of touched on very, very briefly before, is defined by the expression of this BLIMP1 transcription factor, and the expression of that goes up as plasma cells mature. And we were able to take advantage of a BLIMP1 GFP reporter mouse, which was generated here at WeHi, to look at plasma cell responses throughout infection and also a long time after. And so first I looked at the total plasma cell response, and that's sort of looking in this gate here, which we defined as BLIMP1 positive, CD138 or syndican positive cells. And I found that at day 6 post-infection here, that the frequency of that population was was significantly less than in the infected mice compared to the immunized controls, and that sort of evened out at later time points. And we found that reflected when we calculated the total number of those plasma cells, and it appeared as though infected mice had hardly any plasma cell responses at all. So one of the advantages of the BLIMP1 GFP model is that you can actually begin to differentiate the two different populations of plasma cells. So we can specifically identify short-lived plasma cells by their low expression of BLIMP1 GFP, and we can identify the higher ones by the higher expression of BLIMP1 GFP. And so I looked at the frequencies of those populations in my infected and my immunized mice, and consistent with what I found before, the frequency of the short-lived plasma cell population, was reduced in infected mice compared to the immunized controls. And when I looked at the frequency of the long-lived plasma cell population now, we found that that was fairly similar between the two groups, which is not uncommon when we use the BLIMP1 GFP reporter mouse. So we wanted then to sort of get an idea of the quality of that plasma cell response. And because we aren't able to study antigen-specific cells during a malaria infection just yet, I used IgG1 as a surrogate marker for that. And I found that, that isotype switching to IgG1 was really significantly impaired in the infected group compared to the immunized control group. And that was both in the short-lived and the long-lived plasma cell population. And so when we take all that data together, we see that, that not only is the short-lived plasma cell population really inefficiently generated during a severe malaria infection, but both the short-lived and the long-lived populations have limited isotype switching, which may indicate that they're slightly less efficient during infection. And given that especially these long-lived plasma cell populations are thought to emerge from the germinal center response, that led us to hypothesize that the germinal center response may be impaired during severe malaria infection. And to begin to characterize that, I looked at splenic architecture by confocal microscopy. So this is just what a naive mouse spleen looks like in the steady state. So you can see the T cells stained in blue and the B cells stained in green, and the germinal center cells are stained in red with antibodies against GL7, and those structures form at the interface between the two zones. So when we looked at day 6 post-infection, at the time of that clinical malaria episode, we found some really striking differences between our groups, in that those GL7 positive cells would disperse throughout the tissue in the infected mice, but also that the T cells had become depleted from the T cell zone and had begun to infiltrate the red pulp. On the other hand, when we looked at our immunized mice, we found that germinal centers had already quite robustly formed within the naive B cell follicles, and the T cell compartment had remained abundant. However, we found that as we analysed the response as it got further away from the clinical episode, that germinal centres could actually form in the infected mice, as you can see by these small structures. And the further and further away you got from that clinical episode and all the accompanying inflammation, the more the germinal centres between the two groups became indistinguishable. And so given that germinal centres require the interactions between different subsets of cells to occur, we decided to take a closer look at those subsets by flow cytometry, and we began by looking at the B cells within those germinal centres. And we defined those cells as CD19 positive B cells, which express low levels of CD38 and GL7. And we found that, consistent with the imaging data, that the frequency of that population was significantly reduced in the infected mice compared to the immunized controls. We found a similar difference at day 9, and that sort of begun to even out around day 12 post-infection. And you can see that reflected when we calculated the absolute numbers of those cells, in that population was induced with much slower kinetics in our infected mice. I then went on to look at the T follicular helper cell population, which was that second germinal center population, and we defined that as activated CD4 T cells, so that express low levels of CD62L, with high expression of PD1 and CXCR5. And we found that, once again, similar to the germinal center B cells, that population was at a much lower frequency in the infected mice compared to the immunized controls. However, once again, as we got further away from that clinical episode, that population did start to appear, although to a lesser extent, and finally by day 24, the frequencies were similar. And once again, we found that reflected when we calculated the absolute cell numbers of those populations and found that the T follicular helper cell population was induced with much slower kinetics in the infected mice. So more recently, a second T follicular helper cell population has been defined, and that T follicular helper cell population appears prior to the induction of the germinal center response. And it also expresses lower levels of the common T follicular helper cell markers such as CXCR5. And as a consequence is sort of termed a precursor T follicular helper cell or pre-T follicular helper cell population to the fully mature T follicular helper cells. And with more analysis, we were able to define that population in our infected and immunized mice, and we found that it was at similar frequencies between the two groups, all throughout the response. And that was reflected when we calculated the absolute cell numbers of that population. So when we take the data on the T follicular helper cells together, we see that even though at day 6 post-infection, which is when that really strong clinical episode is happening, even though at day 6 post-infection, infected and immunized mice have similar sized pools of pre-T follicular helper cells, the emergence of the fully mature T follicular helper cell population has occurred much more slowly in the infected group compared to the immunized ones, which might suggest that there's a differentiation defect somewhere in the T follicular helper cell lineage. And we were able to sort of get a better idea of that by looking at the ratio of these pre-T follicular helper cells to fully mature T follicular helper cells, so just dividing this number by that number. And when we plotted that out in a graph, we found that in infected mice, the ratio sort of was quite low and approached 1. So if we think that one pre-T follicular helper cell is becoming one T follicular helper cell, then we would expect to have low ratios of those precursor cells to the fully mature ones, which is what we see in the immunized mice. On the other hand, in the infected animals, that ratio was much, much higher, suggesting that there had been limited differentiation of the T follicular helper cell population in those animals at day 6 post-infection. And that ratio dropped off once the mice had been fully drug cured, but it still remained significantly higher in the infected animals throughout the response. So we needed to confirm that these cells actually belong to the T follicular helper cell lineage. Fortunately for us, the T follicular helper cell lineage is defined by 2 major factors. And the first of those is the cytokine IL-21. And that's secreted by those cells and is really important for maintaining an effective germinal center response. And the second is the transcription factor BCL6. Now, transcription factors come into play when during a sort of T helper lineage commitment at the time of antigen priming. So, for instance, in the case of malaria infection, which is a highly inflammatory response, you see a really strong induction of T helper 1 type responses, and that's characterized by the induction of the transcription factor TBET, driven by the transcription factor TBET. And so in the same way, BCL6 drives the specific function of T follicular helper cells in that it's required for the upregulation of markers such as CXCR5 as well as the cytokine IL-21. And BCL6 is upregulated quite rapidly following antigen stimulation, and so you find it expressed in pre-T follicular helper cells as well as T follicular helper cells. So we were first able to take advantage of an IL-21 GFP reporter mouse, which was generated here at WeHi, and we first looked at the fraction of cells, CD4 T cells, which express IL-21, which sit in this gate here. And you can see that the frequency of that population is higher in the infected mice, but when we quantified those 2 populations, they were of similar size. When we went on to look at PD-1 and CXCR5 expression on those gated cells, we found a similar result to what we saw in the wild-type animals, in that there was this accumulation of intermediate expressing PD-1 and CXCR5 cells in the infected mice, but in the immunized ones, a high population was much more discernible, sort of lending credence to the fact that those T cells were belonging to the T follicular helper cell lineage. However, IL-21 is also secreted by other T helper cell subsets, which we weren't able to exclude at the time of this experiment. So I went on to look at BCL6 positive CD4 T cells, which sit in this gate here. And so when we analysed that population once again for PD-1 and CXCR5, we found similar types of population distribution, again with this accumulation of intermediate expressing cells, which further confirmed that those cells belong to the T follicular helper cell lineage. Now, although BCL6 is is considered to be the master regulator of T follicular helper cell differentiation, it's becoming more and more clear that T follicular helper cells can integrate other environmental signals to allow for a measure of flexibility in their responses. And in particular, in the early stages of T cell activation, the balance between TBET, that transcription factor I mentioned before, and BCL6 can become very important in determining the final commitment. So that in highly inflammatory environments, the balance can be tipped in favor of TBET expression and function, which leads to sort of a more predominant differentiation of T helper 1 type effector cells over T follicular helper cells. So given that we knew that severe malaria infection was a really highly inflammatory condition, we decided to take a look at the expression of TBET in our BCL6 positive cells. And what I found was that TBET was expressed almost 50% higher in those cells in infected mice compared to the immunized controls. And I found similar results when we broke down those populations by PD-1 and CXCR5. Now, TBET drives the expression of its own functional markers, and it can, for example, transactivate the expression of CXCR3, which, as I mentioned at the beginning of my talk, is upregulated during severe malaria infection and draws those cells to inflamed tissues. And so, given this high TBET expression, we looked at CXCR3 expression in our BCL6 positive CD4 T cells and found that almost 100% of those expressed CXCR3 in the infected mice, compared to about 70% in the immunized ones. And this also happened when you broke down those populations by PD-1 and CXCR5. And so this suggested that the T follicular helper cells generated during infection might have a really strong T helper 1, T follicular helper cell type dual phenotype. And so in summary, for this section, I showed that when we compared that infection with exposure to a same amount of attenuated parasite, the germinal centres were significantly disrupted by the severe malaria infection, along with a more generalised disruption of splenic architecture. I found that T follicular helper cells were being sort of inefficiently generated from a pre-T follicular helper cell pool, but also that those T follicular helper cells expressed really strong signatures of a T helper 1 type response, and those strong signatures may have been leading to poor commitment of those T follicular helper cells to the T follicular helper cell lineage, and thus why we didn't see effective differentiation. So in order to investigate that a little bit more, we began to look into the ways in which the inflammation induced by severe malaria infection might be modulating all those germinal centre and T follicular helper cell responses. And so as I mentioned at the beginning of my talk, one of the effects of that really strong inflammation is to upregulate inflammatory trafficking pathways. And to remind you quite quickly of those pathways, what happens is that T cells and NK cells will upregulate CXCR3 expression. And concurrent with this, gradients of CXCL10 will be established towards the brains of the infected mice, causing those cells to begin to migrate and infiltrate the tissue where they'll contribute to pathogenesis. And importantly, when CXCL10 is neutralized, either with specific antibodies or using genetic deletion, there is less infiltration of those pro-inflammatory cells into the brains of the infected mice, which leads to enhanced survivability of those animals. And in addition to this, CXCR3 is still upregulated, but those cells are retained within the spleens where they can participate in more robust immune responses. And so because we saw really high CXCR3 expression on our T follicular helper cells in infected mice, we hypothesized that those T follicular helper cells might be trafficking away from the spleens and into the brains, and thus not able to participate in the germinal centre response. And to assess this, I infected CXCL10 deficient mice. And so when I infected those mice, I found that consistent with published data, there was enhanced retention of CD4 T cells in the spleens, which led to measurable increases in the size of the pre-T follicular helper cell population, and to a lesser extent, the T follicular helper cell population. But that when we looked at the germinal centre response, that was not actually improved. And so that suggested to us that migration along that CXCR3, CXCL10 axis was contributing to a minor depletion of the T follicular helper cells from the spleens of infected mice, while not actually changing their function in any way. So For instance, the fact that CXCR3 upregulation still occurs in these CXCL10 deficient mice might indicate that the underlying inflammatory responses that are leading to that induction are still occurring, and that's what might be impeding T follicular helper cell differentiation. And so with that in mind, we went on to look at the cytokines that are known to play a role during T follicular helper cell differentiation. And the two main drivers of that are IL-6 and IL-21, which can act via different STAT molecules to upregulate BCL6 and induce the T follicular helper cell program. However, depending on the model used, other cytokines have also been found to intervene in these processes. And in particular, interferon gamma and IL-12 can favour the upregulation of Tbet over BCL6 and lead to this enhanced T helper 1 type differentiation. And in addition to this, IL-2 is known to directly inhibit BCL6 function via interactions with STAT5 and other transcription factors. And more recent data suggests that type 1 interferons can also play a role in this process, and depending on the model used, they can either promote or inhibit the differentiation of T follicular helper cells by different STAT molecules. And importantly for us, we know that a number of these cytokines are upregulated during severe malaria infection, and that, for instance, interferon gamma is known to be a really important driver of pathogenesis, and type 1 interferons have also been shown more recently to have a role in that process. And that led us to hypothesize that those pro-inflammatory cytokines that were being induced during severe malaria infection might be inhibiting T follicular helper cell development and effective germinal center responses. So surprisingly, given the higher IL-12 responses that I measured in the infected mice at the very beginning of my talk, the infection of IL-12 deleted mice did not improve the T follicular helper cell or the germinal center B cell response. And we found a similar result when we infected interferon alpha receptor mice, which have no type 1 interferon signaling. However, when we went on and we infected interferon gamma deficient animals, we found that there was a significant increase in the T follicular helper cell population and a trend towards an enhanced germinal center response. Now, as I mentioned quite a while ago at the beginning of my talk, interferon gamma is not the only pro-inflammatory cytokine that's known to play an important role during severe malaria infection. And TNF has also been described to participate in that severe disease in both humans and in mice. And in addition to this, interferon gamma and TNF are known to interfere with splenic architecture in other disease models. So in order to extend these observations to include TNF, I treated infected mice with specific antibodies against TNF and interferon gamma. And infection of those mice led to an increased frequency of the T follicular helper cell population at day 6 post-infection, which then was accompanied by a significant increase in the size of that, the absolute size of that population, as well as an increase in the germinal center B cell population. And then when we looked at the splenic architecture of those animals by confocal microscopy, I found that the T cells were sort of far better retained in the spleens of the anti-cytokine-treated mice than they were in the isotype control-treated mice. And that it appeared as though aggregates of the GL7 positive germinal center cells were forming earlier. And we saw that reflected at later times following infection, where germinal centers were more abundant in the anti-cytokine-treated mice compared to the isotype control ones. And so that suggested that the pro-inflammatory cytokines produced during severe malaria infection were impeding the induction of T follicular helper cells and germinal center responses. And to get a better handle on how that differentiation was being impacted, I came back to look at the ratios of those cells across all the experiments. So just to remind you that a low ratio, as we see in the immunized mice, sort of indicates efficient differentiation of pre-T follicular helper cells into T follicular helper cells, and we found that there was a very high ratio in the infected mice. When we looked at the CXCL10 deficient animals, that ratio had dropped down almost to half the level of that seen in infected mice, suggesting that this pathway was contributing to the depletion of fully mature T follicular helper cells from the infected animals. But more significantly, when we looked at both interferon gamma deficient mice or anti-cytokine treated mice, we found that ratio was significantly more tipped back in favour of T follicular helper cell differentiation, suggesting that those cytokines were playing a role in impeding those processes. And so while we were able to exclude a role for IL-12 and type 1 interferons in T follicular helper cell differentiation during severe malaria infection, we did find a role for interferon gamma and an additional potential role for TNF. And so what emerges from all of this data is this picture in which interferon gamma in particular may be mediating poor germinal center responses and poor T follicular helper cell differentiation in a number of different ways. So as we saw in the CXCL10 deficient mice, interferon gamma dependent inflammatory trafficking pathways were contributing, such as the upregulation of CXCR3 and CXCL10, were contributing to a depletion of the available T follicular helper cell pool in the infected animals. And then by using the interferon gamma deleted mice, we were able to see that interferon gamma may have been having a slightly more subtle effect, more on a sort of genetic level. And although we haven't yet tested this, what might be happening is that interferon gamma is promoting the strong upregulation of Tbet, which then leads to inhibition of BCL6 function and then ineffective T follicular helper cell generation. And a third possibility, and one in which TNF may also be playing a role, is in disrupting splenic stromal cell and possibly antigen-presenting cell populations. So the start of the T follicular helper cell differentiation programs requires really specific types of cognate interactions between T cells and antigen-presenting cells. And if the splenic architecture is disrupted by pro-inflammatory cytokines, those interactions may not be occurring efficiently enough to lead to strong activation of the T follicular helper cell program, once again leading to ineffective T follicular helper cell differentiation. And so to begin to investigate this a little bit more, in the very near future, I will be using an adoptive transfer system in which we take IRF4 floxed CD4 Cre recipient mice, and IRF4 CD4 Cre recipient mice have defects in the activation of their CD4 T cells, and more importantly for us, they lack fully differentiated T follicular helper cells. So we take out these mice and we give them one dose of irradiated parasites, and the next day we sort out cells from donor mice which have been pre-infected or pre-immunized with Plasmodium berghei sort of 6 days prior. And those activated CD4 T cells are then sorted and adoptively transferred into those mice. And we can assess the immune responses at later times following the adoptive transfer. And so using this transfer system, we hope to look at two questions concerning T follicular helper cell biology. And the first of those is whether or not the T follicular helper cells that are generated during severe malaria infection are permanently imprinted with that sort of T helper 1 T follicular helper cell phenotype that we saw, or potentially via mechanisms like epigenetic changes, or whether or not that phenotype is far more flexible and only really depends on the immediate environmental cues. And the second thing we hope to be able to study is whether or not those T follicular helper cell populations have any different functions. It's been proposed in the literature that pre-T follicular helper cells are more efficient at supporting short-lived plasma cell responses, whereas the T follicular helper cells support the germinal center reaction. And so we hope to be able to see whether there are any differences in those functions in between our infected and immunized mice. And I've been able to perform preliminary experiments using this system, just looking at PD-1 and CXCR5 expression on the activated CD4 T cells before and after transfer of those cells. So as you can see here in the infected mice, as we've found previously, there's very few PD-1 CXCR5 expressing cells. But when those cells are transferred into an environment which is clear of severe malaria infection, that PD-1 and CXCR5 begin to upregulate, which sort of suggests that whatever break exists during severe malaria infection is lifted, and that those T follicular helper cells can go on to differentiate properly, which also further lends support to the notion that severe malaria infection is impeding the generation of T follicular helper cells. So that brings me sort of to the end of my talk. And in conclusion, we've shown that by using a model in which we can compare active severe malaria infection to exposure with the same amount of antigen, that severe malaria infection impedes the correct generation of plasma cell responses, but also that it disrupts the germinal center response, which could, and also in the context of more generalized splenic architecture disruption. Finally, we also found that limiting those inflammatory processes that were associated with severe malaria infection helps to rescue T follicular helper cell differentiation and also improves germinal center induction. Finally, we found that once the severe malaria episode had been cleared by anti-malarial treatment in the mice, the germinal center induction did actually occur, which would suggest to us that our model is similar to what might happen in a low transmission setting with the person who experiences the one clinical episode and then goes on to develop immunity. And so we would like to extend this model to be able to study what happens during multiple infections in a high transmission setting. So in this setting, an individual would get a malaria infection and potentially develop severe malaria symptoms. They would be taken to the nearest treatment centre and given anti-malarial drugs. And after that, would probably have induction of germinal centres as we see in our infected mice, and induction of memory B cell populations. However, we know that in really high transmission settings, rates of reinfection can be really quite high. So with the next malaria infection that individual experiences, that will lead to dissolution of ongoing germinal center reactions, which will effectively undermine the generation of completely protective immunity. And so although this remains to be tested using our model with multiple reinfections, if it holds true, it could have really important implications for effective anti-malarial vaccine development. So that's the end of my seminar for today. So I thank you for your attention, and all it remains for me now is to thank everyone who had a hand in this project. So I want to thank Diana for giving me the opportunity to work on this project and also for giving me a lot of freedom to do a lot of exploratory research. I thank my co-supervisor Axel for coming on sort of halfway through and having to suddenly deal with malaria infection and everything that entails, and also for giving us a lot of mice and support and advice. I want to thank my PhD committee, particularly Alan for his support, and the Infection and Immunity Division as a whole, because it really is an absolutely fantastic place to work. And in particular, I'd like to thank in my lab, Lisa and Anne for their hands-on support. From the Schofield lab, Emily and Danica for helping me to design different fax panels and also for troubleshooting. I want to thank Simon from the Pellegrini lab for helping me with some mouse work and also giving us the BLIMP1 CD19 Cree animals. And I need to express my immense gratitude for the molecular immunology and immunology divisions, because without a lot of their resources, much of this work wouldn't have been sort of possible. So Steve Nutt and Julie Tellier for the R21 GFP and BLIMP1 GFP reporter mouse, David Tarleton and Lynn Corcoran for some mice and some experiments that I didn't show today, and Dimi, Kirsty, Kim and Melinda for various protocols, reagents and help. I want to thank Warren Alexander for the interferon gamma knockout mice, Liz Hartland and Andrew Brown from the Doherty for the IL-12 knockout mice, and Paul Herzog and Kelly for the IFNA knockout mice. And so given that this project was very, very mouse work heavy, I want to thank all the people in bioservices who've helped me out, particularly Liana, because I probably couldn't have done half the experiments without her help. And she's also happy to lend an ear when you need a whinge. As you could tell that this was a fax-heavy project, I thank Simon and the Fax Lab for their hospitality, for helping me with the sorts and for occasionally helping me to fix the machines when they inevitably broke down. I thank the imaging facility for their support. Kelly and Lockie and Cameron, who's now left, were really, really important in helping me set up and optimize a lot of the stains. I want to thank instrument services, Dennis and Tom, because obviously without irradiated samples, I would have had no project, more or less. I want to give a thank you also to Wendy Carter and Joan Curtis, because Plasmodium berghei is a quarantinable parasite, which means it doesn't exist in the wild. And it means that we have to jump through hoops, the likes of which only a government department could come up with, and they help us to comply with everything so we can keep on doing our experiments. I finally want to thank my friends, particularly the ones that we hire, because it's no small thing having a group of people who understand what you're going through, who can listen to you when things go wrong, and who can celebrate with you when things go right. I want to thank also, very end, family and my partner Phil, for their unending support, patience, advice, and cups of tea in the face of all the craziness that comes with having a PhD student for a daughter and a girlfriend. And thank you to you guys for your attention today.
Thanks, Victoria, for a lovely talk. Questions? 10 minutes, plenty of time.
Nothing?
Yes.
I guess my question is going to the innate immune side of things. So that initial burst of gamma interferon, is that coming from NK cells, or do you think it's all T cell mediated?
No, so that initial burst of interferon gamma is known to come from NK T cells primarily, I think, and then that will sort of start off everything going. So it's not, I mean, I don't think it's ever been dissected out the exact sort of sequence of events. But yeah, I know that NK T cells, are a really big source, and so are NK cells, and that'll sort of be the trigger for all the priming of the T cells and everything that follows on from that.
Yes, the first thing I'd like to congratulate you. Beautiful work,
Thank
huge
you.
amount of work that you put in. It's really nice to see how it all comes together.
Thanks.
But I'd like you to draw back a bit from your responses to two observations we see in humans that are different from your mouse system. The first thing is you showed quite clearly that while you've shown in your system that severe malaria severely impairs the acquisition of memory responses, we know that immunity to severe malaria is acquired in nature very rapidly. 1 or 2 infections, even on the high transmission, or very few infections, the risk of severe malaria drops very dramatically, while it takes much, much longer to get immune to clinical malaria. The other observation is that when people get exposed to malaria as adults, they have a much higher risk of severe malaria in the first place, but they get immune much faster than children. And those two observations, how do they fit with your observation of severe malaria impeding the memory formation?
So, I mean, I would suggest that in our model at least, we wanted to look closely at memory cell induction following our model. And we weren't really able to do that because what happened was that, as you saw the germinal centers being induced, you did actually, and well, preliminary data that we've got would suggest that the memory cell responses are actually induced at the same rate between the two groups. And that's probably because there are germinal centers forming, and that confounds the results. That made it very difficult for us to be able to actually look at this effect of severe malaria infection on memory cell populations. So I don't think that it's I don't think that the two sort of effects that you see in nature, in humans, where severe malaria immunity is induced quite quickly, and what you see in our model can be are exclusive of one another. So I don't know enough on the mechanisms that protect you from severe malaria and the ones that protect you, that lead to those protective responses as an adult to really be able to solidly comment on why that's different. Yeah, sorry. I don't know if Diana has a comment on that point.
Well, I would just go back to trying to extrapolate Victoria's results, what we know about human malaria. And the truth is that we know very little about T follicular helper cell malaria in human malaria. But one of the things that is starting to emerge in studies by Peter Crompton's lab in Mali is that individuals with acute malaria have follicular helper cells with a funny phenotype exactly like the one that Victoria found, with low expression of CXCR5 and PD1 and poor helper capacity. So I think that if we basically try to concentrate on the research of this thesis and try to extrapolate that into human malaria, isolating the follicular helper cell component, I think is very similar. Danica.
Correct me if I'm wrong, with the CXCL10 knockout mice, you saw, in the wild type, you saw more migration of the pre-TFH cells to the brain?
We didn't measure direct migration. So I never took the brains of those animals and looked for T follicular helper cells of infected mice. But in the CXCL10 deficient mice, what you find was this accumulation of the pre-T follicular helper cell population and a slightly smaller accumulation of the T follicular helper cell population. So that's what led us to think that the CXCR3 expression on our T follicular helper cells was drawing some of them, but not all of them, away from the spleens of the infected animals.
I guess my question is, do you think those pre-TFH cells might be contributing to cerebral malaria in the model?
So that's a question that we would have to look into. So I guess one of the... So those as we saw by our IL-21 GFP mice, all of those cells sort of were producing IL-21. And IL-21 is known to sort of modulate inflammatory responses. So I think that it can I'm not sure if it can sort of further prime CD8 T cell responses to become more inflammatory. So I guess in that respect, there is a potential for IL-21 secreting pre-T follicular helper cells to be contributing to pathogenesis in the brain, but whether that's true or not, would have to look into that, I think.
Just to make a short comment on that, the main population contributing to cerebral malaria in this model is CXCR3 positive CD8 T cells, not CD4. So if you look at the composition of the brain sequestered leukocytes in these animals, the vast majority are CD8 T cells. But there is a component of CD4 T cells, like 30%, which we never looked at it, as Victoria said, but they could be expressed in CXCR5 and PD1, but it's a minority.
Just as a matter of, it might be naive, did you take the look at any other time points past the 24 days? Like, did the trend continue, for example?
No, I didn't. I think, yeah, I didn't do any facts like later than day 24. In Shabauti models of disease, when they looked at sort of germinal, they only looked at GL7 positive B cells in that model, and they found that those cells could be detected out till like day 50, I think. Which would suggest that, once that parasite is clear, enough antigen remains in the system for immune responses to actually occur against that. But yeah, I don't know how long those would go out for in my mice.
Hayley.
That's a very good question. When you're looking at the effect of the cytokines and you have the interferon gamma knockouts versus the ones treated with antibody, did you have any where it was just the TNF? Like, so do you know what the contribution of TNF was, or was it always anti-TNF and anti-interferon gamma together?
Yeah, so it was always both neutralizing antibodies together. We never got to we never actually managed to do this two separate neutralization studies. So we thought about using sort of TNF knockout mice for a while, but they have a lot of disruption to splenic architecture, even in the developmental process, so that kind of excluded us from being able to use those properly. And yeah, we just kind of never got to the point where we did sort of the two separate animals. And we thought that there's been, in the Bergii model of cerebral malaria, there's sort of been a more strong point made for interferon gamma in mediating the responses rather than TNF, which no one's really ever described a really strong role for that in Bergii malaria. But I mean, that sort of data suggests that it is having some role somewhere. We just don't know at what point yet.
It's a very expensive experiment.
Yeah.
Probably a naive question, but I think you mentioned somewhere in your talk that the disruption of the germinal center induction has been seen in disease contexts outside of malaria? And whether anything could be learned from that?
Yeah, so it's not specifically germinal center reaction, so I haven't actually been able to find any other infection models where they've specifically looked at germinal centers and how those are induced throughout the infection. What has been seen is that during leishmania infection, TNF mediates loss of marginal zone B cell populations, and then that contributes to overall loss of splenic architecture in that model. And I think also in leishmania infection in dogs, it was found that that infection could impede the expression of the homeostatic chemokines that are required for the maintenance of splenic architecture. But whether that's by TNF or interferon gamma, that wasn't looked at. And then interferon gamma, I think, can interfere with populations of stromal cells during LCMV infection. So I mean, that sort of just suggests that those cytokines have a role in disrupting splenic architecture rather than specifically disrupting germinal centres. And that's why what we think is happening is that sort of three-pronged approach, that you've got disruption of splenic architecture, and on top of that you've got other more specific effects of the cytokines on the cell differentiation themselves, plus the migration.
One last question, or two. Simon.
Is it possible to do successive rounds of drug cure and reinfection to see, can you get like acquired immunity?
So we haven't actually tested it out. So one of the, I think we have to play around with the model a little bit, because what we find sometimes is that if you infect a Bergii mouse, and then you reinfect it and you drug cure it, and then you reinfect it with another really high dose of parasite, they actually tend to get quite sick a little bit faster. So I think, but I mean, as with all models, it sort of requires a bit of adjustment and sort of working out. But I think that we haven't tested it yet. We have, when we really, like, it's sort of something that's been thrown around for like the whole course of my PhD, and I just haven't, other more interesting things kind of propped up. So I
that one like tease out a bit more what's sort of required.
Yeah.
I was just going to pick up on the TNF versus interferon gamma question. It just occurred to me when that question was asked that, well, there are millions of people out there on anti-TNF therapies for various things, and hopefully they wouldn't be allowed to stay on that therapy and go to malaria and endemic areas at the same time. I was wondering if we know anything specifically about germinal center reaction splenic architecture under that kind of anti-TNF therapy.
Oh, that is something that I have not looked into at all. So I can't answer that question. I'm really sorry. I mean, TNF is required during the development of secondary lymphoid organs to sort of maintain that, to even induce that architecture. And so generally I think the effect of TNF is sort of only sort of really occurs in a steady state during organogenesis. But when you just treat a mouse with anti-TNF antibodies, it doesn't sort of change the architecture, which is kind of what we saw in our experiments, where the architecture was better in our treated mice compared to the isotype control treated mice. So maybe it wouldn't have much of an effect at all, just because their organs, I mean, if it's in an adult, their organs would be fully formed, and the populations that the TNF acts on, which I think are like the more sort of stromal cell populations, that it doesn't have that effect because they're not sort of being generated.
All right, we need to finish. Please join me in thanking Victoria for a fantastic talk.